Hello, folks. Welcome to the first talk from the Physio Webinar 2022. And thank you very much for being here today. This is an event organized by students from the Physiology graduate program of the University of São Paulo. This is our team. For this season, we organized 3 amazing talks with speakers from different countries talking about different themes of physiology and their intersections with other areas. Please follow us to get more information, okay? So today we are very glad to welcome Dr. Jeffrey Iliff. He's the Associate Director at the VA Puget Sound Healthcare System. He is also a professor in the Department of Psychiatry and Behavior Sciences at the University of Washington School of Medicine. Dr. Iliff's research has focused on the brain vasculature as the crossroads of CNS. As a postdoc at the University of Rochester Medical Center, he led the theme that probed the lymphatic system, a brain-wide network of perivascular spaces that facilitates the clearance of waste products, including amyloid beta and tau from the brain interstitium during sleep. Since starting up his own lab in 2013, Dr. Iliff's work has focused on defining the glial and vascular changes in the aging and post-traumatic brain that underlie impairment of lymphatic function and the vulnerability to the development of conditions like Alzheimer's disease, Parkinson's disease, and chronic traumatic encephalopathy. Dr. Iliff, thank you very much for your presence here today. After your talk, we'll have 30 minutes to answer the questions from the audience. Everyone can ask questions in the YouTube chat. They could be in English, in Portuguese, or in Spanish if you like, guys. This the event will be recorded and will be available through YouTube after the transmission. Now, I will give the word to Dr. Iliff, and I will be here in background in case of any technical problems. Enjoy the lecture, guys, and see you soon.
Very good. Thank you, Kevin, for that kind introduction. And thank you to the Physio Webinars team for the opportunity to present today. It's exciting to be able to talk in front of a global audience, and also as the parent of teenagers to actually get to be a YouTube star for at least just a minute. So I'm going to be providing a little bit of a whirlwind tour of what has come to be known as the glymphatic system, which is the brain's waste clearance system, which we described initially only 10 years ago, so back in 2012. So over the last 10 years, work in this space has fundamentally transformed how we think about fluid movement in the brain and how that fluid movement and changes in it underlie the development of diseases like Alzheimer's disease. So if you are interested in learning more about my lab, here's a QR code. If you scan it, it will take you to my lab's website where you can see the other things that we're doing. And if you are a QR code person, you'll note that some of these are going to come up throughout the talk. So if you're interested in a specific topic that I'm going into, you can follow the QR codes to lists of references that will give you more information. So I first need to start out with a couple of disclosures. So my lab is funded by the United States National Institutes of Health, the US Department of Defense, and the Department of Veterans Affairs. I serve as the chair of the scientific advisory board for a company called Applied Cognition and as a consultant to Griffin Biosciences. Their work is related to glymphatic function, so keep that in mind as you listen to my talk today. I'm also a full-time US federal government employee. And so all of whatever you hear from me today is my opinion and not the opinion of the United States federal government. So I wanted to start just with a little bit of a cue into the lens through which my lab views physiology. So our lab focuses on the causes and consequences of neurodegenerative diseases. So these are dementing disorders like Alzheimer's disease or Parkinson's. We are interested most particularly in the ways in which they're similar to each other. And one of the ways that one of the things that links all of these different conditions is the fact that each of them is characterized by the misaggregation, so the clumping up of different proteins and peptides in brain tissue. So in Alzheimer's disease, its histological hallmarks are the presence of extracellular aggregates made up of the peptide amyloid beta and intracellular aggregates made up of the protein tau. In Parkinson's and in Lewy body dementia, what you see are inclusions or aggregates inside of neurons made up of the protein alpha-synuclein. So one of the things my lab evaluates is what is it about the brain that makes it vulnerable to this process of protein misaggregation? What is it that drives these common features of different neurodegenerative conditions? In particular, we're interested in how different risk factors contribute to these processes. So those risk factors include aging, cerebrovascular disease, exposure to traumatic brain injury, and sleep disruption. So we asked the question of what changes in the aging brain or in the injured brain that makes it vulnerable to these processes of protein misaggregation. So to start, I'm going to talk a little bit about cranial fluid dynamics, which is maybe something that you never thought you'd be interested in, but I think it ends up being important for thinking about the biology that we're talking through. So cranial fluid dynamics, we're talking about fluid or water movement inside the brain. So if you've learned anything about cranial fluid dynamics, which most people haven't, you've learned maybe something about cerebrospinal fluid circulation. So cerebrospinal fluid, or CSF, is a clean, relatively protein-free and cell-free fluid that flows through the ventricular system of the brain. So that's a series of interconnected caves within brain tissue. So it's secreted by the choroid plexus, which is a secretory epithelia hidden inside these ventricles. It flows out by pulsatile motion through the ventricular system, out into the subarachnoid space, which is the space that surrounds the brain. And from here, the CSF is reabsorbed thought to be reabsorbed through structures called arachnoid villi into the big venous structures that surround the brain. And so the idea is that there's this one-way circuit of fluid motion from the ventricles out into the space that surrounds the brain and into the bloodstream. Now the things classically, so from about the 1700s up to just about 10 years ago, the things that we thought that CSF did was that it helped to float the brain, it helped to support it under its own weight. It helped to cushion the brain from impacts or blows. It helped to balance volume within the brain. So when blood flow increases in one part of the brain, one of the ways that volume change within the skull can be offset is by changing the amount of fluid, the amount of CSF, either in the ventricles or in the CSF. And it was thought that the CSF serves as a sink, or as a place where wastes from brain tissue get deposited. And so the CSF sink hypothesis, which goes back into the 1960s, it was based on a couple of things. It was based on an anatomical observation that the CNS, so the brain and the spinal cord, they lack lymphatic vessels. So in the periphery, lymphatic vessels help to serve the clearance of interstitial fluid, so the fluid between the cells, and the reabsorption of proteins from the spaces between cells. It contributes to immune surveillance, so the ability of the immune system to detect things that are happening. And so the CNS, since it lacks lymphatic vessels, it was thought that the CSF serves as a sink for these proteins and for these wastes, and it helped us subserve immune surveillance. And the way that it did that was that fluids and solutes from throughout brain tissue would wander their way slowly and diffusely out into the CSF that surrounds the brain or into the CSF within the ventricles. And that this process, which was slow and diffuse and not physiologically regulated, was how the brain took care of the process of waste clearance. At least that's how we thought that things went until about 2012. Now, if you are in the neuroscience space, you may know that major pieces of this hypothesis have come into revision in just the last decade. So in 2015, two different groups, one in Virginia in the US and one in Finland, showed that contrary to what most people thought, there are in fact lymphatic vessels surrounding the brain. They're not in the brain, but they're in the tissues in the meninges, so in the membranes that surround the brain, immediately adjacent to the brain and spinal cord. These meningeal lymphatic vessels seem to participate in the clearance of solutes, both from brain tissue and from the fluid that surrounds the brain. So this is a major new finding that totally changes how we think about waste clearance and immune surveillance in the CNS. Second, this notion that the fluid flow between the CSF surrounding the brain and the brain interstitial fluid within the CNS, the notion that that process is slow and diffuse and not physiologically regulated, well, that turns out not to be true either. So this is an experiment here that demonstrates the movement of CSF around the brain that is both rapid and anatomically organized. So in this experiment, this is in a mouse. We're doing 2-photon microscopy, which is a way for us to do fluorescence imaging down into the brain of a living mouse. We've injected a red tracer into the bloodstream so you can see the blood vessels. So these are arteries and veins at the brain surface, and then these are blood vessels down within the brain tissue itself. We inject a green fluorescent tracer into the CSF that surrounds the brain. And what you can appreciate is that this CSF tracer, it doesn't just sit on the outside of the brain, but rather it follows very specific anatomical pathways. It moves initially along these surface arteries, and then as these arteries dive down into and through brain tissue, you see that this green CSF follows along. And this green haze that you see out in the brain tissue is actually CSF that isn't just sitting outside the brain, but is rather washing into and through brain tissue along the outsides of blood vessels. So this isn't something that is just happening here microscopically at this little tiny part of the brain. This is actually something that's happening everywhere. So this is a very similar experiment here, except that we're using a rat instead of a mouse. And we're using MRI instead of fluorescence imaging to measure the movement of fluid and solutes from the CSF surrounding the brain into and through brain tissue. So this is using a very powerful MRI magnet. So a normal magnet that you would use for like a clinical MRI would be either 1.5 or 3 Teslas. This is a 12 Tesla magnet, so it's very powerful. We inject a contrast agent, in this case it's gatoteridol, into the CSF that surrounds the brain. So we're illuminating the fluid around the brain. And then what I've done is I've encoded the signal change that you see in the first hour in blue, the signal change in the second hour in green, and the signal change in the third hour in red, so you can appreciate how things happen. So here what we see is that the CSF surrounding the brain, it initially moves along the ventral surface of the rat's brain. We can see it down here in the coronal slice. But we can also see that its initial movement into brain tissue occurs along blood vessels. So this line right here, that's the anterior cerebral artery. This line here, these are the choroidal arteries. These lines back here that we see on this axial cut, these are the posterior cerebral arteries. So the movement of CSF into and through brain tissue is occurring along the axis of the brain-blood vasculature. So it's actually using the blood vessels as a scaffold to organize the movement of fluid into and through brain tissue. This isn't something that's only happening in mice and rats, it's also something that's happening in people. So this is the first of a series of experiments that were done by a group in Oslo in Norway, where they did essentially the same experiment that I just told you about in a rat. They did the same thing in people. So this is a study in 8 humans, so they're around 40 years old. These were being evaluated for neurosurgery, so there was a reason why it was justified to inject a contrast agent into the CSF that surrounds their brains. They injected the same contrast agent, and then they scanned the people over time. And what you can appreciate is the contrast agent, which shows up in white here, it initially surrounds the brain, enters the CSF spaces surrounding the brain at its base. It begins to move up around the front and eventually begins to enhance the wider brain tissue. When the participants came back the next day, so after 24 hours, what you can see is that the entire brain is enhanced relative to what you saw the day before. What that means is that this contrast agent, which is showing the CSF, is actually moved into and through the brain tissue of these human participants, showing that there's this rapid movement of CSF into and through the brain, not just of mice, but also of humans as well. So to summarize, about 10 years of physiological work that's been done in mice and rats and primates and humans into one slide, we now know that this perivascular movement of fluid, which we call lymphatic exchange, and the clearance of solutes by these lymphatic vessels embedded within the membrane surrounding the brain, those are meningeal lymphatic vessels that drain the CNS, we now know that this process is anatomically organized and rapid. We know that it's driven by arterial pulsations and the pulsations of blood vessels. We know that it is supported by astroglial water transport, so by water transport through the brain's support cells. We know it appears to be a feature of the sleeping rather than the waking brain, which is an interesting physiological thing. And we know that these two processes, perivascular exchange through brain tissue and meningeal lymphatic drainage, these are actually coupled physiologically to each other. You shouldn't take my word for it. You should never take anyone's word for anything. You should see the data yourself. So if you follow this QR code, it will take you to the papers, to a list of the papers that tell us that this is true. All right, so I'm going to go a little bit more into detail into how it is exactly that we know a couple of these things. How do we know that this process is supported by water transport through the brain's support cells, astrocytes? And how do we know that this is a feature of the sleeping rather than the waking brain? So first, I'm going to give a real fast overview of how we understand the relationship between sleep and Alzheimer's disease. Now, Alzheimer's disease, as I mentioned before, it's characterized pathologically, meaning in the tissue itself, by two key features. The first is the presence of extracellular plaques made up of amyloid beta. So these are these round globs that you see here. These are amyloid plaques, and intracellular aggregates of tau called neurofibrillary tangles. If you don't have plaques and tangles, you don't have Alzheimer's disease, right? So this is the key feature of the disease. Now, it's long been appreciated that there's a close and intimate relationship between sleep disruption and Alzheimer's disease. So it's well known that people sleep more poorly as they get older. And it's known that people, when they become demented with Alzheimer's disease, they have very, very profound impairment in normal sleep-wake regulation. In fact, it's typically the disruption of the normal sleep-wake cycle that drives a person out of the home care environment into institutionalized care, because the family caregivers just can't take care of a person who's up all night and sleeping all day. It's too tough on the family. So it's long been assumed that the relationship between sleep disruption and Alzheimer's disease results from the fact that as the brain degenerates, the brain circuits that regulate the sleep-wake cycle themselves are degenerating. And that's why dementia and neurodegeneration are coupled to Alzheimer's disease. Now, more recent data suggests that the association and the causation may go the other way, that poor sleep may actually be driving the process of amyloid and tau deposition and neurodegeneration itself. So this was an interesting study. This came out of a lab in St. Louis in the US back in 2019. It was a study that was done both in mice and in humans, but I'm showing you the kind of the key human finding here. So what's happening here is the investigators are measuring amyloid levels. So this is the peptide that makes up these plaques in the CSF that surrounds the spinal cord of humans through the course of a whole day. And what they observed was that there's a diurnal, meaning different during the day and during the night, a diurnal fluctuation in amyloid levels in the fluids surrounding the brain, with increasing levels through the daytime and declining levels overnight. So what this suggested was that something about the sleep-wake cycle was actually regulating levels of amyloid beta in people even before the onset of Alzheimer's disease. So there's some close relationship between amyloid levels and what's happening with them and the sleep-wake cycle. Another series of studies, and this is a kind of a key one from them, this was published in JAMA Neurology back in 2013, suggested that there was a relationship between how people slept and the quality of their sleep and how many of those amyloid plaques were present in their brain. So this is a study not in people who are demented, but in people who are cognitively normal. So these are normal people. They're older, but they're not cognitively impaired. And they evaluated in these people, how much do they report sleeping on a normal night? Do they sleep more than 7 hours a night? Do they sleep 6 to 7 hours a night? Or do they sleep less than 6 hours a night? And what they observed was that people who reported sleeping short periods, so in this case, less than 6 hours a night, or what I'm not showing here, people who report poor sleep quality, even when they're cognitively intact, they have higher levels of amyloid beta. And here, amyloid plaque burden is being measured by positron emission tomography. So you can measure how much amyloid is in the brain of a living person in this way. So what this data suggests that there is a very tight relationship between sleep disruption and the presence of Alzheimer's related pathology, even before the development of cognitive impairment and dementia. So these emerging data suggest that sleep disruption, especially disruption early in the aging process, so during midlife, seems to be an important driver and risk factor for the development of the pathology underlying Alzheimer's disease. Now, this is a big statement, right? You shouldn't take my word for it. You should go check out the literature yourself. So if you follow this QR code, it will take you to a series of studies that has begun to show this relationship between sleep disruption and the development of Alzheimer's disease. Okay. So if sleep disruption is associated with Alzheimer's disees, what do we know about this waste clearance and this fluid dynamics that I just told you about? How does that vary with sleep, or is it regulated by the sleep-wake cycle itself? So this is a similar experiment to what I showed you before. So this is the same kind of imaging where we're imaging CSF tracer movement down into the brains of mice. Here we've injected a 3 kilodalton, so kind of a peptide-sized tracer into the CSF. And we're doing two different experiments. One where we're imaging in an awake mouse, and it looks like maybe my video isn't moving, or maybe it's frozen, but it's not. It's just that not much tracer goes into the brain of an awake mouse. But then if you take that awake mouse and you anesthetize it, so this is the same mouse in two different settings, in two different conditions, you see that the situation completely changes. If we quantify the movement of CSF tracer into and through the brain of a mouse, either compared between an awake state and an anesthetized state, or of course, anesthesia isn't the same as sleeping. So we can also compare naturally sleeping mice and awake mice. We see that this washing of fluid through brain tissue is dramatically different between the waking state and the sleeping state, with much more rapid fluid exchange and waste clearance in the sleeping compared to the waking state. Now, this isn't just CSF movement through brain tissue. If we measure the actual clearance of stuff out of the brain, we see that the same thing is true. So in this experiment, we can inject a radioactive tracer into the brain of a mouse. So with a radioactive tracer, we can measure it with very, very high sensitivity. So in this case, we inject either an inert generic molecule, in this case inulin, or we can inject amyloid beta, and then we can measure over time how fast it gets cleared out of the brain. So this is the clearance of inulin, the inert tracer. This is the clearance of amyloid beta. From these clearance curves, we can calculate a rate constant that tells us roughly how fast are things getting out. And what you can see is both for the generic molecule and for amyloid beta, when we compare clearance in the waking animals compared to the naturally sleeping or the anesthetized animals, it's much more rapid clearance in the sleeping compared to the waking state. So what this tells us is that in the sleeping state compared to the waking state, the brain has shifted into a mode that supports the rapid exchange of fluid through brain tissue. Again, it's important to know that this isn't just something that we see in mice. This is something that's happening in humans as well. So this is a study that was published out of that from that same group that I told you about in Oslo a couple minutes ago, where they're doing these same kinds of studies, but in people. This is a really cool experiment. It's a really good experimental design. So what they did is they took two groups of people and they injected gadolinium contrast agent. So that's an MRI contrast agent into the CSF that surrounds the brain. And they measured how much of the contrast agent loaded into the brain in the first 6 or 7 hours after injection in both groups. And what you can see is between the two groups, there's about equal levels of loading. But then what they did is one group they allowed to sleep normally overnight, and the other group they kept awake all night. So they underwent a whole night's sleep deprivation. And what you can see is that the gadolinium in the sleeping people cleared at a certain rate in the first night and in the second night. But in the people who were kept up all night, there was a slowed clearance. There was a slower, the clearance of that solute out of the brain tissue was slower with sleep deprivation. In fact, the difference between them, even when these people were allowed to sleep on the second night, it didn't catch up until several weeks later. So it took more than one night for the clearance deficit to catch up. So that means this waste clearance that's happening in the brain during sleep is happening in the human brain as well. So if this lymphatic exchange or this waste clearance that happens during sleep participates in the clearance of things like amyloid beta, could the impairment of waste clearance actually be contributing to the vulnerability of the aging and the injured brain to the development of things like Alzheimer's disease? So we're going to go through a series of studies where we start to evaluate this question. So first, we're going to talk about changes in lymphatic function in the aging and the injured brain. And we're going to focus first on the aging brain. So question one is, as animals or people get old, does waste clearance change? And this is a super simple experiment. We measure this fluid exchange in healthy young adult mice. So these are sort of 2 to 3 month old mice. So these are, 20-year-old mice in human years. So maybe they're making bad life choices and kind of running around doing crazy stuff. These are middle-aged mice here, so about 18-month-old. We've injected in this case 2 different tracers, 2 different sizes, 2 different colors. And what you can appreciate is in the healthy young mice, there's pretty rapid movement of CSF tracer through brain tissue. In the aged animals, you see a really marked slowing of CSF movement through. So there's a big reduction in how much of this fluid exchange is happening. If we measure the clearance of solutes like amyloid beta, we see that the same thing happens. So in the healthy young adult mice, we see that in the first hour after injection of radioactive amyloid beta, about 70% of the amyloid gets cleared. In middle age, so these are 12-month-old mice, and in old, those are 18-month-old mice, we see a stepwise slowing in the clearance of amyloid out of the brain. So as the animals get older, there's less CSF movement through brain tissue, and there's less amyloid clearance out of brain tissue. So this process of waste clearance has slowed. Now, those are data from aging animals. Over the last 10 years, a large number of studies in animals and an increasing number of studies in humans have now demonstrated that there's an impairment of this waste clearance in traumatic brain injury, in ischemic injury, so like strokes and small vessel disease, with subarachnoid hemorrhage, cortical spreading depolarization, mouse models of Alzheimer's and Parkinson's, mouse models and rat models of hypertension and diabetes. Many of these are risk factors for the development of Alzheimer's disease. We also see in human participants evidence for the same sorts of things. Again, don't take my word for it. I'm just putting words on a screen. I could write anything down here that and you wouldn't know the difference. So go check out the papers. So go to this QR code, and you can go check out all the studies that have been done in the last 10 years, showing that there's an impairment of this process in the setting of injury in addition to aging. So if this perivascular exchange is impaired in the aging and in the injured brain, what is it that underlies those changes? What is driving the changes in this clearance process? So our lab over the last 5 to 6 years has focused on one particular molecular process that I'm gonna kind of focus on for the remainder of my talk. Now, the caveat here is we don't think that this is the only thing that's changing, but we think it is one of the things that's changing. So I'm going to focus on. We focused on the function of something called aquaporin 4. So aquaporins are a family of proteins that span lipid membranes. So they cross the lipid membrane and they form a pore that allows water to transit the lipid membrane easily. So there are many different aquaporins. In the brain, there's two aquaporins mainly. There's aquaporin 1, which is in the choroid plexus, where it's involved in CSF secretion. And then there's aquaporin 4. If we look at how aquaporin 4 is localized, or where it is in the brain, here it's labeled in purple, you might think it's mainly in the blood vessels, but that's actually not true. It's actually mainly in the astrocytes, which are these star-shaped cells, support cells within the brain. It's mainly in those support cells, but what it is it localizes mainly to the part of those cells that faces the brain circulation. So it's part of these cells called perivascular end feet. So these astrocytes send feet that ensheathe the brain blood vessels, and a huge amount of those end feet are actually occupied by this water channel. So we think that this water channel, which is perivascular in its localization, we think that it actually helps to support the exchange of fluid through brain tissue along these perivascular pathways. So the way that we test whether aquaporin 4 is involved in this waste clearance, one way that we can test it is we can look in a mouse where the gene that encodes the aquaporin 4 protein is disrupted. So these mice don't make aquaporin 4. And then we can measure what is the effect of the loss of that gene and of its protein on this waste clearance process. So in this experiment, we inject a green tracer into the CSF that surrounds the brain, and we measure how much of the tracer moves through a mouse brain in 30 minutes. In an animal with aquaporin 4, we see that it's pretty extensive and pretty rapid. In animals that lack aquaporin 4, we see that CSF movement through brain tissue is markedly reduced. We can also measure the clearance of solutes out of brain tissue, and we see the same thing. So here we're injecting two different tracers, again, a generic small solute, mannitol, and amyloid beta. We inject it into the mouse brain, and we measure how fast these things are cleared over the first hour or two after injection. And what we can see is that mannitol and amyloid are cleared in normal mice in blue here with a certain kinetics at a certain rate. So in healthy young animals, we see that about 70% or so of the amyloid is cleared in the first hour. And we see that in animals that lack aquaporin 4, the clearance both of a generic tracer and of amyloid are markedly reduced. So what this tells us is that aquaporin 4 is an important part of, plays an important role in supporting the washing of fluid through brain tissue and the clearance of waste out of brain tissue. So then, could it be that changes in aquaporin 4 are underlying the impairment of this waste clearance that happens in the setting of aging, or that happens in the setting of injury? So maybe aquaporin 4 is down-regulated. Maybe there's less of it in the old brain. It turns out that that's not true at all. Our hypothesis was that there would be less of it, but what we observed was that actually there's more. So if we measure how much aquaporin 4 there is in the old compared to the young brain, here we're using a Western blot to measure aquaporin 4 expression, we see that expression goes up. But the interesting thing is that while expression goes up, and so this is immunofluorescence, where you can see in the healthy young brain, most of the aquaporin 4 is along blood vessels. So that's what all these little lines are, or brain blood vessels. We see that while there's more purple in the aged brain, it's not just perivascular. So it's not just the blood vessels. If we look at an individual astrocyte, we see that in the healthy young brain, most of the aquaporin 4 is along this blood vessel here. But we see in the old animal, it ends up spreading out over all of the fine processes. So we think that what's happening here is as the aquaporin 4 mislocalizes, so as it moves away from the blood vessels to these other processes, the brain loses its ability to organize fluid movement along blood vessels. We have a transgenic mouse that helps us get at that question really directly. So we know a little bit something about aquaporin 4. We know that it is perivascular because a series of proteins link aquaporin 4 to the extracellular matrix at the blood vessel. So there's aquaporin 4, alpha-syntrophin, dystrobrevin, dystrophin, and dystroglycan. It's a series of proteins that link together, anchoring aquaporin 4 at the perivascular input. If you delete any one of the genes in the genes encoding any one of these proteins, whether alpha-syntrophin or dystrophin, aquaporin 4 localization goes from here in the normal animal, where it's mostly along the blood vessels, to this, where there's still aquaporin 4. It's just everywhere. It's all over the cells. So we can use this animal as a tool to test whether does loss of perivascular aquaporin 4, like we see in the aging mouse, does that impair perivascular exchange? And does that promote the development of Alzheimer's-related pathology? So this is a contrast-enhanced MRI study. So it's the same kind of study I showed you before. This is in a mouse. We inject a contrast agent into the CSF that surrounds the brain of a mouse. And then we image the mouse 30, 60, and 90 minutes after injection. And what you can appreciate is that there's this enhancement that happens through brain tissue as the CSF tracer moves through. We can compare a normal animal that has perivascular aquaporin 4, and we can compare it to an animal that lacks perivascular aquaporin 4. And we can see that there's a marked reduction in the rate of fluid movement through brain tissue. So that tells us that it's not just that the brain needs aquaporin 4 to be there, it has to be at the right place. It has to be along the blood vessels for lymphatic function to work. We can also cross this gene deletion into a transgenic mouse, so a genetically manipulated mouse, that naturally forms amyloid plaques, just like in the human brain. So this is a genetic model of Alzheimer's disease where the mice naturally make amyloid plaques that are labeled here in red, and you can see them here in the cortex and in the subcortical structures. In animals that have alpha-syntrophin and normal perivascular aquaporin localization, we see a certain amount of amyloid plaques here in red, and then we can quantify that in these black dots here. In the animals that lack perivascular aquaporin localization, you can even see just with your naked eye, there's more amyloid plaques in the brains of these animals. And if you quantify it, it's a pretty big difference. So when you lose the water channel from the boundary of the blood vessel, where we think it's organizing fluid movement, you impair lymphatic function and you promote the development of amyloid pathology. So what that tells us is that at least in a mouse, some of what's happening in aging is that there's a change in how the brain organizes water dynamics that impairs this cleaning process, and that can promote the development of pathology, the same kinds of pathology that we see in Alzheimer's disease. But again, who cares what's happening in a mouse? We need to know, is this happening in a human as well? So in the next experiments, what I'm going to show you is studies that we've done to evaluate whether the same kinds of things are happening in people. Is there evidence for this process happening in the setting of human Alzheimer's disease? So this is a similar study to what I showed you before, where we're looking at aquaporin 4 in the cortex, so in the superficial, sort of the top layers of the brain, except we're not looking in mice here, we're looking in people. So these are post-mortem tissue samples from people who died cognitively normal, or with Alzheimer's disease, or with something that's in between that's called mild cognitive impairment, which is kind of an intermediate, just beginning to have cognitive impairment. And when we measure the localization of aquaporin 4 to the blood vessels, so that's this perivascular aquaporin 4 polarization, what we see is that in people with Alzheimer's disease, there's less of that polarization. And people with mild cognitive impairment, which is kind of in the middle clinically, we see that they're kind of in the middle, right? So having less perivascular aquaporin 4 is what happens when you have Alzheimer's disease. In these same people, we can evaluate whether that loss of aquaporin 4 in the astrocytes is associated with their memory impairment, or is associated with more or less amyloid or tau pathology. And what we see is that indeed they are related to each other. So at least in people who are in the earlier stages of the disease and not in end-stage disease, less perivascular localization, so as we move leftward on the curve here, is associated with worse cognitive performance on two different scales. So this is called the mini mental state examination. This is the clinical dementia rating scale. So these are two different tests of cognitive function used in the Alzheimer's disease field. And we see that loss of perivascular localization is associated with worse performance. These are two different scales. These are two different ways to measure amyloid plaque burden and tau pathology. And we see the same thing. Loss of perivascular localization is associated with more amyloid plaques and more tau tangles. So this shows that the same sorts of connections that we see in mice are also happening in the human brain in the setting of Alzheimer's disease. Of course, it's important to note that, that in this study, it could be that this pathology is causing this changes in aquaporin 4. So in a histopathological study, we can only get associations. We can't get a causation. We don't know that these changes in aquaporin 4 are causing cognitive decline or are causing pathology. One way that we can get at that, though, is using genetic studies. So this is a gene association study where we look for whether naturally occurring variations in the human genes that encode aquaporin 4, alpha-syntrophin, or these other elements confer risk to or protection from the development of dementia, evidence of Alzheimer's pathology in the CSF, cognitive decline, or atrophy or shrinkage of the brain. And I'm not going to go into this because this gets real technical and my time's a little bit short. But what we did is we looked at a bunch of different naturally occurring variants in these genes and evaluated statistically whether they're associated with these different features of Alzheimer's disease in a human clinical population. And the quick version of the results are that they are. So 3 different variants in the aquaporin 4 gene, 15 different variants in the distrobrevin gene, 2 variants in the dystroglycan gene, and 1 of the variants in the alpha-syntrophin gene. So variants across this entire structure seem to be associated with markers of Alzheimer's pathology, evidence of cognitive decline, and also shrinkage of the hippocampus and cortex, so atrophy of the brain. So these data, and actually data from several other studies that have now been published, suggests that there is an important relationship between this aquaporin 4 and its associated elements and Alzheimer's disease in human populations. Again, don't take my word for it, please follow this QR code and you'll find 5 different studies that link these molecular elements with the development of Alzheimer's disease in human populations. All right, so a summary and some implications. Summary, the lymphatic pathway, the brain's waste clearance system, right? Brainwashing, but the good kind. It is a brain-wide perivascular network that supports the exchange of CSF outside the brain with the interstitial fluid inside the brain. It appears to be a feature primarily of the sleeping brain. It's impaired in the setting of aging and brain injury, and we see evidence that its impairment may contribute to the development of Alzheimer's disease and other neurodegenerative diseases in clinical conditions. So we think that its impairment in the aging and the injured brain may contribute to the aggregation of different proteins and peptides that may be driving the development of diseases like Alzheimer's and Parkinson's. So we think this may be a common mechanistic pathway linking the development of those diseases. So who cares? Why does that matter? Why it matters is this. So everyone who follows the news knows that there have been a series of failures of Alzheimer's disease-focused therapies. This is a curve that represents the sequence of events in the development of Alzheimer's pathology, in this development of Alzheimer's disease. The last thing that happens, marked here in, I can't tell if that's black or green, I'm a little bit colorblind, is memory decline that lands you in the memory clinic. We now know that a bunch of things happen even before the evidence of cognitive decline occurs. The earliest of which are the deposition of amyloid and tau in the brains of people that occurs maybe decades before the presence of cognitive impairment. So current therapeutic approaches to treating Alzheimer's disease or preventing Alzheimer's disease are targeting people that already have evidence of amyloid or tau pathology. They thus far have proven ineffective, in fact, wildly ineffective. What we now think is that processes that are occurring upstream that are driving the development of amyloid or tau might be better therapeutic approaches for targeting Alzheimer's disease, preventing the formation of this pathology rather than trying to eradicate that pathology once it's formed. So if glymphatic impairment contributes to the development of amyloid and tau pathology, it is to the left. It is upstream of these processes. And so if you could detect glymphatic impairment in people, then that would tell you who's vulnerable to the development of conditions like Alzheimer's disease. And it could tell you it decades before the onset of clinical symptoms, when you still had a chance to intervene and prevent the development of these conditions. Moreover, if you could devise a therapeutic approach that restores glymphatic function, that treats the impairment that we're seeing, then it may be possible to prevent the development of amyloid pathology and tau pathology, thus affecting the primary or the secondary prevention of Alzheimer's disease. It's the same way that we prevent heart attacks by treating hypertension. We may be able to prevent the development of Alzheimer's disease by treating glymphatic impairment in midlife. So with that, my lab, this is the thank you slide to my lab members who do all the work in my group. Our funding through the U.S. Department of Veterans Affairs and Department of Defense, National Institutes of Health. My lab is actively seeking new people, new blood, smart, creative postdocs and students. So if you are interested in what we're doing, go to this QR code. It will take you to my lab's website. We would love for you to reach out to us about opportunities in our group. And with that, I'm glad to take questions as well. So thank you.
So thank you again, Dr. Iliff. Now we have some questions to answer, okay? I will put the questions on the Zoom. The first one is the Zoom. So Henrique Leite is a student and he is an admirer of your career. And he asked, has research advanced regarding the use of the prazosin to improve sleep and collaborate in significantly decrease alpha and beta levels in the lymphatic system during the day and night in patients with Alzheimer's?
Wow. I feel like Enrique has insider information because we have data with prazosin, but it is currently unpublished. So maybe you have a spy in my lab. One of the things that was reported, just for the audience, one of the things that was reported in the 2013 paper that reported the sleep-wake difference was that one of the things that was regulating that sleep-wake difference was norepinephrine signaling in the brain. And so if you gave a cocktail of noradrenergic antagonists, so an alpha and a beta antagonist at the brain surface, you could turn on glymphatic function in the waking brain to levels almost the same as what you saw in the sleeping brain, right? So what that tells us is that waking glymphatic function is suppressed, is kept low by norepinephrine. One of the drugs that they put on the brain was prazosin, which is an alpha adrenergic antagonist. Prazosin is clinically used. It's an old drug used for hypertension. Now it's not really used for that anymore because it's not that good at it, but it's used now for the treatment of trauma nightmares, principally in veterans with PTSD. So we have data that shows that if you give prazosin to a mouse, it improves glymphatic function. We have data in humans that suggests that if you give prazosin to humans, it improves glymphatic function. What we don't know yet, and what we have not yet done, and we have grant funded, so one of the projects that's going to happen in my lab in the next year and a half is we're going to test whether prazosin treatment in mice and in humans changes Alzheimer's pathology levels, at least in the fluid that surrounds the brain. Because it's possible that targeting brain norepinephrine signaling might be one of the ways that you can enhance and restore lymphatic function in aging or after injury. So that's a great question, and it's definitely those are our very next studies that we're going to do.
Thank you, Mr. Henrique, for your question. And now there is another one from John Araujo. Is there any evidence showing a relationship between dysfunction of the gliolymphatic system and mental disorders?
Oh, that's a super question. The simple answer is no so far, but I would say that that's partly a historical accident. So most of the work focusing on lymphatic function in the last 10 years has focused on the subject of, quote, waste clearance. And most of the time when people say waste clearance, what they mean is amyloid, tau, and alpha-synuclein, right? Those are the proteins that aggregate in Parkinson's and Alzheimer's and diseases like that. And so because we have looked at this field through the lens of neurodegenerative diseases, it's like that's the only thing we've thought about. But we know that the same processes that regulate amyloid levels are also regulating probably central neuromodulators. So things like dopamine and serotonin and norepinephrine, which we just mentioned, and probably acetylcholine. These are all molecules that are central to mood disorders like depression or psychiatric disorders like anxiety or post-traumatic stress disorder. So most of us who are thinking about it in this field think that there is probably a dysfunction in the lymphatic system in neuropsychiatric disorders. It's just that no one has yet asked the question, right? And so that's sort of a question that is we're ready to ask now. The field is ready to ask it. We just need the right person to ask it, someone with the right interest, right? And the right access to either animal models or patient populations.
So thank you, John. There is another one from Maiara Moreira. Does the sleepless nights in younger years can in a certain way affect and even slower the process of cleaning and affect the person as the person gets older?
Yeah, so we know something and we don't know something. So the study of lymphatic function is new. So this is the word glymphatic. We made that word up 10 years ago. The word didn't exist anymore 11 years ago, right? So there's a sense where a lot of the studies answering this question, we haven't done it yet. So no one has measured lymphatic function in young people versus old people yet. No one has measured whether sleep impairment early in life impairs lymphatic function later in life. And we don't know how that connects to Alzheimer's pathology. Here's what we do know. When you so there are large cardiovascular studies that were done in the US and around the world over the last 30 years. The ERIC study or the Framingham study are good examples of these, right? These are studies that were initiated to try to define where does heart disease come from? Where do heart attacks come from? But now the people from those studies, they've all gotten old and many of them are starting to emerge out into cognitive impairment. Those studies back in the 80s and 90s and early 2000s asked people about their sleep behavior. And so what we're now learning is that it isn't your bad sleep that you have in your 60s and 70s that is necessarily the thing that predicts whether or not you're going to be demented or not. It's actually the sleep, your poor sleep or your short sleep in midlife. So in your 40s and 50s is the stronger predictor of dementia in your 70s. So it's midlife sleep disruption that is setting the table for late life dementia, right? And so I think that's probably the answer, is that's when the groundwork for these diseases is being laid down. It's midlife sleep, it's midlife hypertension, it's midlife hyperlipidemia, it's midlife exposure and earlier life exposure to concussions, right? That's setting the stage for how these diseases unfold in your 50s and 60s and 70s.
Okay, that's another one from Maiara. Does the same loss of aquaporin happen in CTA? And it could be one more problem to be considered with concussion in athletes?
Yes. So we have two things that we know and one thing that we suspect. One thing that we know is that in animal models, two things we know. In animal models of traumatic brain injury, including mild traumatic brain injury, which is what a concussion is, we see that there's impairment to this process. We see evidence of glymphatic impairment in humans that have been exposed to repetitive traumatic brain injury, right? So this is MRI evidence. We know that CTE, chronic traumatic encephalopathy, that's the dementia that people with repetitive head impacts succumb to, most notably in the American football players. It's called the pathonomic feature, which means the feature that is characteristic of this disorder are tau tangles that are perivascular, right? The key feature in CTE is perivascular tau. So many people think that it's impairment of this perivascular exchange in repetitive head injury that is leading to CTE. As of right now, no one has published data on that. I can tell you that there are people asking that question right now, but that data has not been published yet.
Okay, thank you, Maiara, again. And one of us professors asked a question to Gisele Oda. Thank you for your great talk. Has any difference in lymphatic function been observed between individuals that sleep during the night or during the day?
Not yet. So I think there's two different reasons why a person would sleep in the night versus the day. So there's one setting, which is daytime napping. So those are people who sleep during the night and also a little bit during the day. And at least, so no one has measured lymphatic function and the role that napping plays in it. In terms of cognitive impairment, the data is a little bit mixed on whether napping is good or bad. Some data suggests that napping, the people who nap, especially in old age, have higher risk of dementia. Some data suggests that people who nap have lower risk, and it probably has to do with why you're napping, right? So people who nap because they're exhausted, because their nighttime sleep is terrible, that's probably one thing, versus people who, take a 30-minute or 90-minute nap every day as a part of a healthy sleep cycle. That's probably a different thing, right? So napping is one thing, and no one has looked at that. The second reason why people might be sleeping during the day is because they're shift workers, right? So they may sleep during the day and work during the night, and those people, if they sometimes, especially if they're changing shifts, so they work a day shift for part of the month and a night shift for part of the month. They have a misalignment in their circadian cycle. There is evidence that lymphatic function is regulated by not just sleep-wake state, but also by the circadian cycle, right? And so it was shown that, it was in one of the QR codes. There was a paper in 2020 that showed that there's circadian regulation of lymphatic function and meningeal lymphatic uptake. So what that would suggest is someone who has a circadian sleep-wake misalignment, like jet lag, right? Or someone who's moving between night shift, day shift stuff, one would expect impairment of lymphatic function. But that's an expectation or a prediction. Those aren't data. No one's done that study.
Okay, thank you, Professor Gisele. And the last question is, thank you for your talk. Do you have any data on people who sleeps more than 8 hours a day, and how this may affect this cleansing process? Yeah.
So maybe this is a good one to wrap up on, because it sort of speaks to where we're at in the field. So the answer to all of these questions, if every single question that says, have you measured lymphatic function in this? The answer is no, because the techniques for measuring lymphatic function in humans, they're only a couple of years old. So people are only just starting to ask these questions in human populations. And so at this point, almost all of that data almost none of that data has been published, right? So no one has evaluated all of these different settings, sleep-related settings, looking at human lymphatic function. What we do know is that there are known relationships between these sleep features and the risk for dementia or Alzheimer's disease. And actually, so sleep duration has a U-shaped relationship with dementia risk. So as I showed in the slide with the PET imaging, people who sleep short, so less than 6 hours a night, have a higher risk for Alzheimer's disease and all-cause dementia and higher amyloid levels. The same is true of people who sleep more than, say, 9 hours a night. So long sleepers also have higher risk. Now, what's unclear is whether that risk is because long sleep is bad, or is it because people who sleep long have other comorbid conditions like depression, right? Which is itself a risk factor for dementia. And so it's hard to know whether it's the long sleep or the thing that is associated with the long sleep that's the cause of the risk. But we do know that either short sleep or very long sleep do seem to be not so great.
Let me unmute my mic. So thank you very much, Dr. Iliff, for this great talk. And I wanna ask you a personal question, a simple one, because this topic is not my cup of tea, but I have a question I think you may answer. There is a way to promote the expression of aquaporin in old people?
Yeah, we don't know yet. So it seems like what we see in old people and in injured people is that you actually get higher aquaporin 4. So if anything, expression goes up, but it's just not in the right, it doesn't localize to the right place. And so it seems like localization is the problem. And as of right now, there are two ideas for how localization might be affected that are kind of interesting and kind of new. So I don't know if you're familiar with kidney physiology. Aquaporin 2 in the kidney is regulated. It's a pore, right? The aquaporin's a pore. So the pore itself isn't regulated. It conducts or it doesn't. But the way it's regulated is by insertion of aquaporin containing vesicles into the membrane. Right, and so for example, vasopressin causes the insertion of aquaporin 2 containing vesicles into the membrane in the kidney, and that changes aquaporin 2 function by having there being more of it. There's a group in the United Kingdom led by someone named Rosalind Bill, her and a guy named Philip Kitchen, who have shown evidence in the last couple of years showing that aquaporin 4 in the brain may undergo the same process. And they published a paper in Cell 2 years ago showing that there's a drug called TFP that can regulate that process. So that might be one approach, is regulating that insertion. There's another group at Washington University in St. Louis that just published a paper in Current Biology, maybe. And so aquaporin 4, so transcription translation, right? Mm-hmm. There's a process called translational read-through where the ribosome doesn't honor the stop codon and translation keeps going. And so you end up with a protein that has an extended terminus. Well, it turns out that's something that actually happens pretty often. And there's a bunch of transcripts that undergo it more than others. And it turns out that aquaporin 4 is like the champion of transcriptional read-through or translational read-through. And the extended variant of aquaporin 4, so this paper that was just published, it shows that actually that variant is the perivascular aquaporin 4. So they made a mouse that had 3 stop codons in a row so that it can't undergo read-through, and it doesn't have perivascular aquaporin 4. And what they showed was that animal had that they could they had slower amyloid clearance in that animal model. And then they did a drug discovery experiment where they identified like 3 or 4 different compounds that regulate translational read-through, and hence how much of the perivascular aquaporin 4 there was. And those drugs sped up amyloid clearance, right? So I suspect they have patented those drugs, and I suspect that they are selling them to companies to see if they can do something with it. But those are 2 different approaches to maybe regulating aquaporin 4 localization that you might be able to use in old people.
Okay, thank you. And let's call it a day. All right. So I want to remember for everyone that is a talk tomorrow, and you can saw in our channel. So stay tuned for the next talks, okay? Thank you so much, Dr. Jeffrey, and goodbye, guys.